Hi, everyone, and thanks for attending our symposium. In this talk, I will give you an overview of how you can use graph signal processing in order to integrate the information about brain function and structure. Indeed, you know that from magnetic resonance imaging, just changing the parameters of the acquisition, we can retrieve two types of information in particular that we are interested on. One is the functional piece of information, so from functional MRI, regarding functional activations in the brain. And the other is the structural information from diffusion MRI that allows us to virtually reconstruct the white matter bundles that are physically connecting different regions of the brain. So what we aim to do is actually to combine these two pieces of information, which is not trivial as they come from different acquisitions. And graph signal processing offers us new means, a new framework to do this integrated analysis in a non-trivial way. In fact, we consider the structural connectivity, so the anatomical wiring of our brain, as a graph, and the fMRI signal as a graph signal, meaning a signal that is defined on the very same nodes of the underlying structural connectome. Then on top of that, our signal, since it's an fMRI time course, also changes in time. But what we consider as graph signal is an fMRI frame, so a brain spatial pattern of fMRI bold signal at one time point t. And then we will repeat the analysis for each time point. And the question is, how is the function using the underlying structure? So the first step of the method is the eigendecomposition of the structural connectome. So here, for example, you have a structural connectome and regions by n regions. What we can do is eigendecomposing the Laplacian, and in order to obtain structural harmonics, which are represented by the eigenvectors of, in this case, the Laplacian, or it can be the adjacency matrix, for example. So in parallel to what Katarina presented about functional harmonics, so these have more or less the same meaning, but they don't come from the functional connectome, but from the structural one. So they contain information about how the brain is wired. Each of these patterns, each of these structural harmonic is related to a spatial frequency, lambda, which is represented by the associated eigenvalue of each eigenvector. So more in detail, this is the real structural connectome averaged of 100 HCP subjects that I considered in my analysis, and for an atlas of 379 RIs from the Glasser parcellation and subcortical. And this is the plot of the normalized Laplacian eigenvalues from this structural connectome. So this is how it looks like. So you see that the first eigenvalue will always be 0, and then we have increasing values. So what does it mean that these values are spatial frequencies? We can see it, we can interpret it by having a look at the associated eigenvector, so what we call structural harmonic. The first one is a constant pattern, and in fact it corresponds to the eigenvalue 0, which is the direct, the DC of our decomposition, let's say. So we can interpret that, we can explain that as no spatial variation, no spatial frequency, so spatial frequency 0, no spatial variation across the brain. When we look instead at low frequency eigenvalues, these correspond to patterns that are global, as you can see here, and with low spatial variation. What it means is that you have patterns that are very smooth on the graph, on the structural connectome. They are very attached to the structural connectome. In particular, connections or regions that will be very connected in the structural connectome will end up having very similar values in these patterns, in the structural harmonics. So you see that these patterns capture the main axis of variation in the structural connectome. For example, left to right, anterior to posterior, and combination of those. But for sure, what we can say is that by construction then, these patterns are very coupled with the structural connectome. They are they contain a lot of information of the SC. On the contrary, when we move towards the other end of the spectrum, so towards high frequencies, we have patterns with a very high spatial frequency. So where you have most of the time very focused patterns and a lot of variation that do not relate anymore much to the structural connectome. So you have patterns that are detached, more decoupled from the structural connectome. So understanding these differences between between low and high frequencies in the spectral domain of the structural connectome is very important from what comes next. Because in fact, the second step is the decomposition of the functional activity, so one fMRI frame at one time point, with respect to the structural harmonic. So in other words, we take one fMRI frame and we explain it as a linear combination of structural harmonics. So we estimate these coefficients as hat that correspond to the weight of each structural pattern that is present in the fMRI signal at each time point. Then the third step, so after functional activity decomposition or translation into the spectral domain of the structural connectome, the third step is the filtering of this functional signal. So the functional signal, let's say S1, so the frame at time point T1, is splitted into two components, one that only includes low frequencies and one that only includes high frequency structural harmonics. So this is a real example. You see that this corresponds to subdividing each fMRI frame into two portions, one low frequency portion and one high frequency portion. But since we said that low frequency patterns were more coupled to the structural graph, then the first portion, we can define it as more coupled, where the function is more coupled to the structure, while the second portion of fMRI signal will be more decoupled. This translates into the coupled portion really reflecting what are the structural axes, for example. And you see here clearly a posterior to anterior gradient, while the decoupled portion including more the portion of the signal that rides on top of it, let's say, that is not justifiable anymore, or not justifiable 100% with the underlying structural architecture. So we do it for each time point, this decomposition into low frequency and high frequency, and what we find is therefore two time courses, one that is coupled and one that is decoupled from the structure for each node of the brain. And then what we aim is to quantify this coupling and decoupling for each node. And so in order to compute a metric, a global metric of coupling and decoupling across time, we compute the L2 norm across time of coupling of the coupled and decoupled time courses for each node. And then the ratio between this decoupling and coupling is what we call structural decoupling index, which is therefore a node-wise metric quantifying the degree of decoupling from the structure of the fMRI signal, so at each node. But then what we can do is also consider edge-wise metrics of structural function coupling. To do that, basically what we want to do is finding equivalence of the functional connectome. So normally from the time course, the normal fMRI time course, you can find pairwise correlations. So a functional, what we call functional connectivity. Here we want to find an equivalent of that, but from the coupling and the decoupling, from the coupled and decoupled portions. And so we find equivalence of the FC, a coupled version of it and a decoupled version. So what we will call, what we refer to as CFC and DFC. So here we can summarize the full pipeline. We have first a decomposition of the structure to find structural harmonics that are purely structural. Then we take the functional signal, we decompose it in terms of this structural harmonics, which is what we're doing is a graph Fourier transform to take us in the spectral domain of the graph. And then we filter these fMRI frames in low frequencies and high frequencies, so in two portions, one coupled, one decoupled from the structure. And then as a fourth step, we compute node-wise and edge-wise metrics of structure function coupling. So at the node level, the structural decoupling index, SDI, at the edge level, two versions of the functional connectome, one coupled and one decoupled to the structure. Then let's have a look at the results. So what we found in previous work is that the structural decoupling index is not constant across the brain, but indeed it has a meaningful spatial pattern going from more decoupled regions in the high level cognition regions to more coupled ones in the sensory regions that you can see here in blue. We confirmed also the behavioral relevance of this pattern, but with a neurosynth analysis where we related topic terms to different masks, so with different levels of SDI, of structural function coupling. And what we found was a gradient from coupling to decouple from lower level behavioral domains to higher level ones, a gradient that is much close to the one that is well known in literature already, but based on the functional organization, so which has nothing to do with the structure. And then we can interpret this spatial pattern also based on previous literature on other studies that have nothing to do with brain function, or fMRI or function structure coupling. One example is genetic studies that found very similar patterns to our decoupled and coupled network, but based on genes that are used for neural transmission. In particular, they found that the network that for us is coupled uses genes for faster response, while the decoupled network uses genes for a slower response. So we can imagine that when the brain needs to act fast, so in the case of stimuli regarding primary sensory networks, then it follows the structure. So it takes the faster route, let's say. But indeed, when we don't have instead a temporal constraint, then we can imagine that the brain can afford also taking undirected paths that are not related to the structure anymore. Then the question that we asked ourselves is, okay, can the structure function coupling reveal more than a group level organization, such as we showed so far? In particular, how does it look like in different individuals or in different tasks instead of resting state? So in particular, can it characterize individuals? So can it perform fingerprinting, what is called fingerprinting, in basically differentiate, recognize an individual only based on the structural function coupling? Or can it differentiate different tasks? And so to perform, to investigate these two different questions, we actually computed a structure function coupling also on 7 tasks for the same cohort of 100 HCP subjects. And we performed classifications to see if we can correctly classify tasks, so for task decoding, and if you can differentiate subjects, so for fingerprinting. So first, we can see that the brain patterns that are related to subject and task effects that we can retrieve with a 2-factor ANOVA, where one factor is subject and the second factor is task, they're different. In particular, the task decoding brain pattern is more related to unimodal, so it includes more unimodal patterns, so primary sensory regions, while the second one, the subject fingerprinting, is more related to bigger patterns that have include also regions that we know being more related to fingerprinting. And then in terms of accuracies, for both fingerprinting and task decoding, we found very high accuracy, so much higher than the chance level that here would be 0.125. In particular, for node-wise metrics and task decoding, the nodal SDI performed better than an equivalent in terms of dimension, an equivalent measure of functional connectivity. While for subject fingerprinting, what we can remark is that so the accuracies are very high, both for functional connectivity and for function structural coupling, but the highest accuracy is reached by the decoupled portion of FC. So we can interpret that saying that indeed in the decoupled portion of the functional signal that we can find the information that is really specific of different individuals. In fact, this is confirmed also by another cross-validation settings, more difficult indeed, where we chose only one fold, so one specific task for training and one other task for testing in fingerprinting. So for example, we want to identify subjects from rest to emotion, from rest to gambling, and so on. And indeed, even here, the measure that performed better is the edgewise decoupled functional connectivity with near perfect accuracies for almost all the pairs, confirming basically our hypothesis of before that it's in the decoupled portion of the signal that we find this information specific to individual. And the relevance, so the meaning of this information was also confirmed by a behavioral analysis where we related these metrics of structure function coupling and also of functional connectivity with different measures of sustained attention and fluid intelligence with the partial least square correlation analysis. And indeed, we found the highest brain cognition correlation for decoupled FC, as you can see here. And by looking at cognitive and brain saliencies, we can also interpret basically which regions of the brain contribute more to this correlation and which of the scores also are more involved. In this case, we have the sustained attention as one of the mainly correlated, and we can interpret that the higher coupling in red regions of the brain that you can see here, the posterior cingulate cortex, for example, a higher decoupling there is more positively correlated with a sustained attention, while the opposite is true for blue areas, basically. So yeah, as take-home message, we can say that graph signal processing revealed to be a useful tool to integrate brain structure and function, that the structure function coupling is not constant across the brain, but it has indeed the spatial distribution, which is meaningful. It's a well-known gradient and it has a specific behavioral relevance. Then what we showed more recently is that structure function coupling also changes across tasks and individuals. So it performs very well for task decoding and fingerprinting, and in particular is the decoupling that was shown to be particularly important for fingerprinting, being also correlated to individual cognitive traits. With that, I want to thank you, and I wish to enjoy the continuation of this symposium. And of course, I also want to thank you, all members of MIPLAB, in particular, the ones that contributed to this work, Thomas Bolton, Alessandra Griffa, and Dimitri Van de